change affiliations often. I am now at University in Heidelberg, but indeed I will speak about cryo-electron tomography of the whole cell approach, presenting it on malaria parasites. Very briefly, going to the point, we use electron microscope, but the preservation for electron microscopy seems to be critical, namely that we should not destroy the structure before observing it. And this is now believed that cryopreservation or rapid freezing is the method of choice actually to preserve the structure in the intact state. Briefly, what we do, on electron microscopy grid that is seen from a side here, we mount the grid on forceps on device like this. And yes, it is my pleasure to introduce or reintroduce this device here. It's called guillotine as well. Are we in the right city for guillotines? This one is much smaller, and it is made not to kill, but to kill our structure, our sample close to live state. So we do the following. We apply the drop of suspension on EM grid, blot the excess of liquid so that the thin film is formed. And at that stage, we slash everything to the cryogenic liquid and preserve the specimen within milliseconds so that the structures and processes are immobilized within milliseconds and embedded in so-called vitreous water, which is unlike ice that form crystals, so water molecules are oriented. In vitreous water, supercooled vitreous water, water molecules are randomly oriented, like in liquid water. And from the top on electron microscopy grids with holes like this, we see the hole from a side here. We see our specimen embedded in the thin layer of suspension. We can have also the whole cells here or margins of the cells and the like. And as you may know already, this method has been very extensively used in the past 20 years to solve the biological structures. Some of the work is close to atomic resolution, which means that the preservation of the specimen is indeed close to atomic resolution. But to get the structures, the reconstructions from noisy images, one needs to average between a big number of copies of the same molecules, the same size, the same shape, and the same perhaps physiological state. But as you may know, in nature this is not always the case. Very often we see the specimens, even the same virus that is not exactly of the same size and shape to be averaged, not to speak about cells or parts of the cells that are always some differences if you analyze it in details. For that we decided to use electron tomography to get two-dimensional structures of this pleomorphic structure. The principle is easy. A three-dimensional object in the electron microscope generates two-dimensional image. We rotate it slightly, another two-dimensional image, and so forth. 60, 80, maybe sometimes more images in a tilt series that are aligned to the common frame computationally and back projected. Here's projection and here's back projection to a common volume. That's supposed to represent the shape and structure of molecule in quest. There are some distortions on the way. For example, we cannot tilt to full range in electron microscope. That leaves us with kind of living wedge or living cone of information, especially in Z direction of imaging. But also, paradoxically, although we preserve our specimen close to atomic resolution, we can reveal, let's say, one order of magnitude worse only because of that the cryohydrated specimens in electron microscope are very susceptible to beam damage. So with this in hand, we approach malaria parasites with Freddy and Michael in Heidelberg. I'm really lucky that the Plasmodium life cycle has been introduced already by Robert. So very briefly now, not to repeat what he already said, very briefly, we are again interested in mobile forms, in sporozoids that are generated in salivary glands. So salivary gland sporozoids that migrate with own motion, as you already learned in the previous talk. And after injection under skin, they keep moving until liver is infected. To some extent, we also concentrate on non-movable, non-mobile midgut sporozoites. And with the same whole cell approach, we want to approach the other extracellular stages, that means merozoites. And in the other part, the second half of my talk will be our recent approach to the blood stages of malaria. So we recover the sporozoites either by isolating them from salivary glands by squeezing, or by natural salivation through the proboscis here of mosquito. When we place electron microscopy grid here, you see some sporozoids coming out, GFP sporozoids. They walk, this one or a couple of them walking on electron microscopy grid, even more of them in the field. We can correlate later on. We can study the patterns of movements, correlate later on after the guillotine stage to what we'll see in electron microscope. And what we see in electron microscope is shown here. It's one of the cells that you have seen before, now at low magnification, so that we see the whole cell. Very important message now. If we put any other eukaryotic cell, the typical fibroblast, you name them, the bulk of the cell is too thick for electrons to transduct through, leaving you only the marginal part, which is thinner than 1 micron, 0.5 micron, for electrons to transmit through and form the image. And please come back here and look at the spacebar. The thickness of sporozoid at any point is less than 1 micron, making it amenable for imaging and collecting tilt series at any point. And this one, for example, generated 4 tilt series. One of them is seen here, this apical part. When we reconstruct this, well, and you dissect it like you do with baguettes here in Paris. And now the reconstruction is being dissected as we do with baguettes in Heidelberg. Longitudinally, I want to leave you with impression how crowded the environment here is, especially with the vesicular structures, but also cytoskeleton and membranes. Some of them, painstakingly, we are able to trace the continuity of the membranes, find out what belongs to where. This is only practically allowed now when we deal with three-dimensional reconstructions, which was not possible before with only two-dimensional image from electron microscope. Of course, one would expect, and this is truth, we want a tool to systematically, one by one, approach different organelles, the whole morphology of the cell in a more like electron microscopy classical way with this cryo tool, with this three-dimensional imaging. For example, studying localization and shape and structures of nuclear pore complexes, connections, continuations of membranes with, between all these organelles as well. However, what I want to present you now is like a more systematic way of approaching the structure that we see in crowdness of the environment in the cytoplasm of the intact cell. One of the tools, important tools, is called particle identification or template matching. So having the reconstruction, tomographic reconstruction, and a priori structure of molecular machines, cytoskeletal elements, and the like, we scan with these structures through every possible point in every possible orientation through the reconstruction. At the end, fishing out cross-correlation peaks with certain threshold, we can localize, co-localize, find out positions and perhaps functional states of the machine's molecules in quest. If we know the atomic structure, then that means that the atomic structure will be fitted at that high resolution range to our low resolution electron microscopy tomograms of the whole cell. And now at the other side of the resolution, we also want to correlate, forming like a bridge from this atomic unanimated structures and make them in animation of the living cell by correlative microscopy between light and electron microscopy. So again, the cells here, in this case, neurons, anything else, that are deposited on electron microscopy grid, so phase contrast, fluorescence, and the same feature one-to-one correlated to what we will see in electron microscope to win, let's say, 100, 200 times the resolution that we may achieve here. And this we started already on the sporozoites with, this is work practically of Anna Sartori at the moment at the Institute Pasteur. We started this with sporozoites, and if you look at one of them here encircled, many of them are moving, but this one we capture already on this, after freezing, so after this guillotine stage. And Anna put it into so-called cryolight microscopy. And what is this now? This is practically inverted light microscope with a cryo box that preserves, that holds our grid, our specimen close to liquid nitrogen temperature. And then from cryolight microscopy, it goes to cryo-electron microscopy. We zoom in, and at the end, we can also collect information about the three-dimensional structure in tomography. One of the projects here for us is solving the molecular basis of the movement of sporozoites. So we aim on actin, actomyosin. However, this work is in progress at the moment. What is a bit more advanced is the work on microtubules, and we approach them the following way: dissecting one or more microtubules from the reconstruction, which is seen here, unbending them three-dimensionally, and then in Fourier transformation we analyze the layer line, which are layer lines that are structure factor intensities. Actually, the information about periodic structures, periodic because we have, of course, this helical or pseudo-helical symmetry of microtubule, at least periodicity along the axis of 4 nanometers, and we see this 4 nanometers. This is a resolution limit of our reconstruction. And then we filter information that is only allocated to these layer lines at different spacings. We get quite interesting answers from that. For example, if you look, well, here, one of the microtubules directly from the reconstruction, we unbend it, filter it, this one with 32 nanometers striation, and we start to see structural features, let's say these connectors, that are almost completely not seen here. They are not absent, but they are engraved in noise. And when we bend back this microtubule to original shape, put to the original place in the reconstruction, we see that the microtubule have connectors, 32 nanometer spacing, connecting it to this pellicle region, so the set of membranes that surround the sporozoid, the specimen. And that now when we collect more of them, this average more of these structural features of these connectors, perhaps we'll be able to solve the structure of it and learn what molecule it is. So far, this is not known what molecule actually endows this property, and perhaps endows the structure of this elongated cell. Another peculiarity of the microtubules is microtubule end, that in most of the reconstructions we see it open-ended, like a flare, which in higher eukaryotic microtubules, it will mean depolymerization. Here in this case, there is nothing in this system, nothing that recalls dynamic instability. The microtubules are very stable. We assume there must be some other factor which endows stability to the microtubules. And indeed, when we analyze microtubules in more detail, here, these ones, we found out that the wall of microtubule is 3 nanometers thicker than the wall of microtubule in control in higher eukaryotes. But it's made of almost the same tubulin, so it must be something extra. And the something extra is located here in yellow in the lumen of microtubules and not outside. So this is perhaps like a luminal map. And when we take only the central part of like a concentric cable of TV cable or so, only the central part, so the lumen, and analyze it in Fourier transformation, we see the very strong 8 nanometers periodicity along the axis. That means something is in the lumen that interacts with either alpha or beta tubulin monomers or in between them, not with both. Well, we analyze it further, more precisely. If we take only lumen, we see it strongly. If we take only outside, we see almost nothing. We plot this against the radius of microtubule, and yes, we can. The signal colocalizes within the lumen here, actually where the wall starts. Our working hypothesis is this molecule is the one that may endow the stability to microtubules. We analyze it when we crash the sporozoids, also some other systems, tachyzoids of toxoplasma. We always see the same profile of microtubules, this very strong luminal density. And it differs from all the controls that we already had. Except with some exception, to some extent, from neuronal microtubules, in which inside we see luminal densities as well. But these ones of completely different nature, structure, location than those which we see in apicomplexans. It's simply the proof of principle that we can say that lumen of microtubule is not latent, it's not void space, but it could be used for, it could be active, either for transport, but we rather believe it will be for stabilizing, having stabilizing function. And now stability of microtubules, very often imaging in vitro, in vivo, in situ, we see sporozoids bending very sharply. We confirm it in the electron microscope, and when we look at the microtubules in the reconstructions of such sharp bends, we see that microtubules are indeed bending, in this case, to 86 nanometers radius, which is 10 times the curvature that higher eukaryotic microtubules will stand before breaching. So they are truly, truly stable. However, if we go further and we squish the sporozoids by pistons and let them recover, some of them start moving anyway, we see microtubules damaged to some extent as well. And some half an hour later, when we preserve the cells by freezing, we saw the microtubule ends still hanging, pointing towards each other, and some like a cementing factor in between them. What exactly it is, we don't know, but let's assume that these bugs cannot perhaps survive without intact microtubule cytoskeleton. So with these tools in hand, so far we looked at single cells in electron microscope that were rapidly frozen. Now we go to blood stages, which means that parasites, when it invades the red blood cell, there's already a system of the parasite and the host cell. We cannot look at this as a single cell. So the parasite makes itself very comfortable inside and wants to eat, of course, and takes a lot of nutrients from the cytoplasm here of the infected cell, some nutrients from outside. The point, however, is the red blood cell originally has no transport system, and the parasite very smartly organizes the whole transportation, export, import system within the cytoplasm of infected cell, and exports something like 300, 400 different proteins to the host cell. Over this transport system, very prominent feature is so-called Morer's cleft, this lamella or cisterna, the membranous structures that are seen in different representations by different authors here in electron microscopes. Very important factor also is of the export, of the parasite export, that exports through the cytoplasm the molecules, let's say like this PF, EMP1, to the surface of the red blood cell that are responsible for sequestering, for adhesion, aggregation of infected red blood cells in capillaries that causes severe symptoms of malaria. And this is, again, our life. If you look how beautifully the classical electron microscopists may see the Morer's clefts, in cryo-EM we see something like this. It's very faint. And on purpose I show this picture. First of all, ignore these white rings here. There are simply holes in support. The parasite is this darkish structure here, which is inside of the cell, and the margin of the cell is seen here. I don't know if from the back rows you can distinguish that. Here are the structural features of the transport, so the Morer's clefts and the others. But if you take it, if you zoom in, Morer's cleft in this case, and you take it, study in 3D, you start to see beautiful structures. Parasite is somewhere here. Plasma membrane of red blood cell will be there. And let me stop it, wherever. And this is the transport system that is organized by the parasite in the cytoplasm. The Morer's clefts, lamellae, and now from this point, of course, painstakingly we start to study the system. It's very little known. There are different dogmas how the transport is organized. We claim that this native preservation will give us a lot of information anyway. First of all, parasite is somewhere there. Here are these Morer's clefts. We never saw any connection. We've never seen any connection between Morer's clefts and plasma membrane of infected cells. Here is the inner part of the knobs where the adhesive factors are sticking to. But rather at these regions, we see plenty of vesicles. So a working hypothesis is there is a vesicular transport rather than a direct connection to the plasma membrane. And we also start to see, and this is for the first time, actin filaments. So this transport is most likely, very likely organized via actin. And this sheds a new light on the transport system. And not only this, we can also see what is in the beginning of the formation of this Morer's clefts. Also novel observation here is parasite itself, Parasitophilus vocularum membrane has connection, magnified here and then highlighted there, has connection to the cisterna or lamellae of Morer's cleft. And the lamellae are interconnected via tiny funnel-like structures anyway. So it's very often direct interconnection here, perhaps transient. It may appear, disappear, but no connection at the other part. We use other tools now to prove this hypothesis as well. And one of the important tools for us now is immunolabeling, of course. It's not only the structure that we want to look at, but we want to also localize certain sites. And we correlate the structural work of cryo to more classical preservation for electron microscopy. The Tokuyasu sections, for example, here, Stivor, the protein that is very specific for Morer's clefts is labeled, and indeed it colloquializes with Morer's clefts. We were thinking how to combine both the structural preservation and immunolabeling, and we succeeded in one of the first approaches now, that we made small holes in the cells by bacterial toxins, introduced primary antibody, again skeleton binding proteins, and then secondary with gold. And what we got indeed is that this is the first practical combination of immunolabeling with the let me stop at any point, let's say here, with perfect structural preservation. And this is Morer's cleft, not seen classically as you may be used to from this side, but rather seeing flat. That is very strongly and very specifically labeled. So this is one of the first stages now, but we are happy to have this tool in hand. This is practically the same picture as before. Again, with this with hand, the work is not finished, it goes on. We approach yet another, let's say, narrower project now, which is we want to study what actually causes some individuals that suffer of hemoglobinopathies, so sickle cell trait or so, what makes them to be resistant to severe syndromes of malaria. And severe syndromes actually are that the infected cells are aggregated in capillaries to brain, but also in the other systems to placenta. And this is already known that this factor, PFEMP1, among some others, which is responsible for this adhesion in capillaries, is much less exposed in the sickle cell, so the mutation, rather than in infected wild-type cells. This is known, and this is very important information. However, nothing, zero is known how and why it happens. And again, what we want to do, we want to study the whole transport system, whether any differences or not in comparison to the wild types that we already had. And of course, tomography that we employ. Another very interesting tool, what I will briefly introduce at the end, is conditional protein export system, but also everything supported by more classical, like biochemical approaches. And here we see one of the first tomograms that show a sickle cell, so the mutant cell, infected with parasite. And here is parasite itself, here is plasma membrane of red blood cell. And what we see here is, instead of this beautiful classical lamellae of Morris cleft, we've never seen them yet. We see like membranous structures or large vesicles, typically denser than the background. And when we analyze it, again, in this colorful representation now, these are the structure that I just described. Also, what is different is cytoskeleton that is not the same with passing through actin filaments passing through cytoplasm, but rather allocated to the plasma membrane of infected cell. We haven't found any differences with vesicles yet, but we work on this as well. And here, for comparison, is simply the uninfected wild-type red blood cell in the same system that we had before. Very monotonous landscape, a part of the actin spectrin and anchoring close to the plasma membrane. So uninfected cell, infected cell, these are the differences that are caused by the parasite inside the cell. And here, infected sickle cell. This is the last slide, brief introduction to this conditional protein export system. Example is skeletal binding protein, the characteristic protein of the Morris cleft that is tagged to GFP, and then also to this conditional domain that causes aggregation of the whole complex at this point where it is produced. And then externally, when we add anti-aggregation ligand, the export starts, and we have whole population, actually, which is very important for electron microscopy, the whole population of the cells that are synchronized in time. We can harvest, we can stop it at any point by freezing, again this guillotine, and analyze at any stage the export system. So with this, I think I fulfill my duty of presenting to you our novel tools. I collaborate with particularly two groups in Heidelberg, Freddy Frischknecht with Misha, Simone, and Sylvia. Michael Lancer on intracellular stages with Philippe, Nicole, and Stefan. Very operative in the whole story was and still is Anna Sartori, now working at Institut Pasteur. And I collected data on electron microscopes on heavy instruments in Max Planck Institutes in Germany, EMBL, Heidelberg. And for sickle cell, we collaborate with the groups in Ouagadougou, Burkina Faso, and with David in Rome. Thank you very much for your attention.
Thank you, Marek. That was superb. Any questions?
I was struck by the actin that you show in the infected red blood cell. So what do you think this actin does? Is it sort of linking the marrow cleft to the plasma membrane?
Some of the actin filaments we detect there indeed form the link. However, at the moment we cannot prove if the prerequisite for the transport of the vesicles is indeed linked at both sides. However, we see acting like crossing the cytoplasm, especially at the area beyond Morris cleft, so direction direction plasma membrane.
Do you see actin underneath the plasma membrane of the sporozoid itself? Can you locate it, and can you use that to better define the arrangement of the mota?
This is a very important question. We spent now several years, third year now, try to fish out the actin there. Actin is supposed to be recognized relatively easily. However, we don't see it. Our working hypothesis, again, is that actin in sporozoids is in a very, very short form and very perhaps transient. And in vitro, that actin actually polymerizes to maximum like 60 or 80 nanometers, very short. And I think we, from structural analysis, we missed something in this respect.
Do you have an idea? So, to the actin filament you're seeing in the parasite, is that branched, non-branched, or can you comment on that?
You mean bundles of actin? That's
exactly.
That's the question. Seeing where? In parasite or?
In the parasite side, yeah.
Yes. As I said, I mean, in parasite itself, in the whole intact cell, sporozoids, we haven't detected actin. Only when we use drugs, let's say just plaquenil, and then we see bundles. But this is not an objective picture. I mean, this is the effect of a drug.